Oysters are ecologically and economically important. They were introduced to the Pacific Northwest in the early 20th century as a means to replace diminished native oyster populations and have expanded to a worldwide geographic range. They primarily inhabit intertidal and estuarine environments, and they provide high economic value, supporting an industry of $270 million within the Washington shellfish trade. And they also provide vital ecosystem services such as shoreline stabilization, water filtration, as well as habitats for larval and adult fish species. Although summer mortality in this species has been observed since the early 1940s and is a significant stressor and to their survival in the marine environment. Energy demands lead to summer mortality. There's a variety of factors that may contribute to these immune challenges or these challenges that they face in the summer months, such as immune challenges, elevated temperature, energy expenditures that come from growth, feeding, and reproduction. Like I said, all of these increase their energy expenditure and make it maybe less easy for them to respond to environmental stress. Specifically in the Pacific Northwest, Washington shellfish farmers report 20 to 80% of annual losses in harvest, which is obviously a huge economic and subsistence food loss. Obviously, climate change is increasing summer heat stress. This is a photo from 2019 of the blob, a major heat wave that was experienced here in the Puget Sound. This just further highlights how we need to further examine how our marine biota may respond to environmental stress. Oysters are generally tolerant to a variety of environmental stressors such as salinity, pH, temperature, and aerial exposure, just based on where they inhabit. I was primarily interested in the effects of salinity and pH and how they may respond to future stressors. Particularly in the Pacific Northwest, we experience wide variability in salinity through heavy rainfall as well as tidal fluctuations, and this will continue to be exacerbated by climate change. Over here, I just wanted to do a brief overview of ocean acidification. I expect that it's kind of a broad topic, but it's known as where excess carbon dioxide is taken up by the ocean from the atmosphere. In the past 200 years, we've seen a decrease in ocean pH by around 0.1 units, and with different amounts of anthropogenic contributions, our future projections by 2100 further see a decrease of 0.4 units in ocean pH, which is a significant stressor to our marine calcifiers, such as Pacific oysters. I just want to highlight that these stressors, as well as other environmental stressors, may impact their ability to tolerate thermal stress, which is why we see so much summer mortality. Therefore, I really wanted to look into how exposure to low pH and salinity affect Pacific oyster survival under subsequent acute thermal stress. Because of all of these interacting factors, I hypothesized that oysters exposed to either salinity or ocean acidification stress prior to thermal stress would exhibit decreased survival. So, for the first experiment, I used 80 oysters as our sample size, with 40 oysters experiencing control conditions and 40 oysters in salinity stress. These salinity stress experiments were conducted with fresh water in 5-gallon buckets, and they were experimented on for a duration of 3.5 hours to 24 hours with a frequency of 3 to 4 days. This was then followed by 48 hours of thermal stress with mortality assessments at hours 1, 2, 3, 24, and 48. I do want to highlight that this kind of protocol for assessing mortality could be used with any environmental stressor, such as aerial exposure or temperature before their response to thermal stress. Before I go over these graphs, I kind of just want to orient you. On the bottom is going to be hours after thermal exposure, and the y-axis is going to be percent survival of organisms in control in the red and blue treatment groups. For the first trial, consisting of 3.5 hours a day for 3 days, there was no significant difference in treated and control oysters, and this trend was continued throughout the remainder of the trials for 4 hours a day for 4 days, as well as for 24 hours a day for 24 hours a day for 4 days, which was a consecutive exposure for 4 days. I do want you to look at how thermal stress greatly reduced survival, especially at the time points that we measured. After the 3-hour mark, we see that mortality generally increased, and this is obviously very significant as we know that thermal stress greatly impacts their survival in the marine environment as well. So we fail to reject the null hypothesis that prior stress does not have any effect on thermal tolerance, just due to the fact that salinity did not show these differences. The second experiment that I conducted was exposing the same sample size, 80 oysters, 40 for control and 40 for acidification conditions. These were conducted with a CO2 bubbler into a bucket of fresh water adjusted to 25 PSU with instant ocean salt. I do want to highlight that the pH here did reach around 5 to 6, which is an extreme OA stress and not naturally relevant. Although these trials were conducted for durations of 1 to 3 hours for frequencies of 3 to 4 days, again followed by 48 hours of thermal stress with mortality checks at 1, 2, 3, and 24 hours. The plots that I'm going to show you are exactly the same y-axis and x-axis and y-axis as the salinity stress graphs, and control and treatment groups are also the same color. So OA stress did not impact thermal tolerance. We see for 1 hour a day for 4 days, again, that the p-value does not show any statistical significance, as well as with 2 hours a day for 4 days and 3 hours a day for 3 days. Again, I do want to highlight that thermal stress greatly reduced survival. This is a big, obviously significant environmental stressor for them. As well as I wanted you to look at this point over here. When exposed to extreme OA stress at extreme time scales, which was the longest duration of total exposure at 9 hours, we saw at every time point increased survival, a trend for increased survival in treated oysters, which I think is very surprising, just let alone the fact that it was such an extreme OA stress as well. Again, due to insignificance, we fail to reject the null hypothesis that prior stress has no impact on thermal tolerance. For some take-home points, I do want to really nail in that thermal stress is significantly reducing survival in Pacific oysters. In this study specifically, we observed 73 to 87.5 mortality in all oysters exposed to heat stress. And also, compounding environmental stressors have no statistically significant effect on oyster survival. This is interesting, as we would assume that the energy expenditure taken to respond to other environmental stressors such as low salinity or low pH may hinder their response to future environmental stressors, although this was not the case. I do want to also highlight that these results may suggest that Pacific oysters are energetically well adapted to natural variations in the Pacific Northwest, such as fluctuations in salinity and pH. I would like to considerably thank Dr. Stephen Roberts and Ariana Huffmeyer for their guidance, recommendations, and thoughtfulness throughout this research, and I would also like to thank the Jamestown Seafood Point Whitney Shellfish Hatchery for providing Pacific oyster seed utilized within the Roberts Lab. Thank you. Any questions?
So what size were the oysters used in this?
They were 10 to 40 millimeters, so all pretty around this size. Yeah. I didn't want to add too many variables into this. I do have like pictures that I did analyze for like size analysis. I was noticing smaller oysters were dying at a higher rate than those that were larger. But I, again, didn't really feel like that was relevant within this research, but
Answered all our questions.
okay. Thank you.